Hello, everyone, and welcome to today's webinar, Mastering Basic Techniques for Cancer Cell Culture. I am Matt Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. For everyone who is attending the webinar today, we know that understanding cancer cell culture is essential for the life of your work. Gibco Galaxy Tour Webinar has the aim of connecting you to our Gibco experts who will share their knowledge and experience to help you master modeling the complexities of cancer in your research. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Brittany Balhouse, Research and Development Scientist at Thermo Fisher Scientific. Brittany, you may now begin your presentation.
All right, thanks, Matt. Hi, everyone. I'm honored to be able to present some of the basics of cancer cell culture to you today, as well as some of the lessons I've learned from my experience doing cancer research over the years. Here's an overview of the presentation today. First, we'll start with why it is important to study cancer and why there is so much work yet to be done in preventing and treating cancer. Next, we'll go over the hallmarks of cancer and what cell models and tools to consider as you begin your line of research. Finally, we'll dive into how you can use those cells and tools to model the hallmarks of cancer in vitro. And we'll end with some resources that may be helpful for your cancer research. So let's dive in. First, just to ground us a little bit, why is it important to study cancer? Well, although we made some real strides in recent decades in reducing the incidence of some forms of cancer with regulation of carcinogenic materials and vaccinations, for instance, and reduce deaths by catching cases early with screening recommendations, cancer still accounted for nearly 1 in 6 deaths worldwide in 2020. If we take breast cancer as an example, there were over 2 million cases recorded in 2020. Even though we now have a better understanding of genetic mutations like the BRCA gene that can predispose one to breast cancer and have tools like mammograms for regular screening, there were still an estimated 684,996 deaths due to breast cancer in 2020. So why is cancer so difficult to prevent or even cure? Well, cancer is unique to each individual. That is to say, one patient's cancer is inherently different than another patient's cancer. This is because cancer arises from each patient's own cells. You see, there is a lot of cell turnover in the human body, and each time a cell proliferates, there is a risk that a risk of something going wrong with the genetic code. Usually, if something goes wrong and the DNA is corrupted, that cell will self-destruct or be destroyed by immune cells. If the mutation occurs in a very specific gene, however, the cell may not trigger that self-destruction process and may be able to evade immune surveillance. This gives that corrupted cancer cell a chance to divide and acquire new mutations. This may lead to uncontrolled growth of the mutated cells and progression of cancer. In addition to the cell turnover, the series of events may also be triggered by external stressors, as seen in the video here. Here you can see that the external stressor can trigger a point mutation or a chromosomal rearrangement, either of which can trigger tumor growth. So let's next go over the key features of cancer that drive malignancy. These hallmarks of cancer were outlined in a landmark paper by Hanahan and Weinberg in 2000, and they updated and extended their list in another publication in 2011. These papers have become must-reads for new cancer researchers, and I'm happy to report that an overview of these hallmarks can be found in Thermo Fisher Scientific's Cancer Cell Culture Basics Handbook. Now, we've already spoken about a few of these on the previous slide, namely resisting cell death, sustained proliferative signaling, evading growth suppressors, genome instability and mutation, and evading immune destruction. Others include replicative immortality, tumor-promoting inflammation, and evading immune destruction. And the rest we'll discuss in more detail later in the talk. These include inducing angiogenesis, invasion and metastasis, and deregulating cellular energetics. Each of these hallmarks is ripe for scientific inquiry and could be studied singularly or jointly with other hallmarks with cancer cell culture. And of course, the first step to investigating one of these hallmarks with cell culture is choosing the right cell model. There are many things to consider when you're choosing which cancer cells you would like to culture for your research. For example, which cancer type or types are you interested in? Do you need several lines to capture various subtypes within a specific cancer indication? Are you interested in localized cancer, metastatic disease, or the progression from one to the other? What cells have the specific gene or protein expression that you need for testing your potential therapy? It is important that you think through which cancer cells best address your research questions, and remember that several different cell models are often needed. These panels of cancer cells are popular for investigating several cancer types at once, several subtypes within a cancer indication, or to represent patient-to-patient heterogeneity for a given cancer type. This is an example of one such panel. These 4 cell lines represent 4 different stages of breast cancer progression from the same patient. The panel was generated from a non-malignant breast cell line named MCF10A that was intentionally transformed with a mutant HA-RAS gene to create another cell line named MCF10AT. The MCF10AT line models the pre-malignant stage of cancer. The MCF10AT cells were then clonally selected until a line which represented the pure comitoductal carcinoma in situ subtype was found. This line was named MCF10DCIS.com. Finally, the MCF10AT line was also serially passaged through mice until a highly invasive metastatic subtype was established, and the resulting cell line was named MCF10CA1. Thus, the progression from non-malignant to localized disease to metastatic disease, all within a syngenetic background, can be modeled with this cell line panel. Such a panel could be especially useful in seeing how well a therapy is selectively targeting cancer cells, for example. Another thing to consider is whether or not you like to utilize cancer cell lines or primary cells for your cancer research. Studies using cancer cell lines are most common as they provide a few key advantages, namely that they are easy to scale up to get a large number of cells for your study, and you can also culture them for many passages and don't have to keep isolating cells from tumor tissue every few weeks. There is also a huge publication record that you can reference to help with your study and to contextualize your research for publication. However, the downside is that cancer cell lines, which have been in culture for decades, can be hypermutated as compared to freshly isolated tumor cells, and thus no longer represent the patient tumor from which they were derived. Primary cells, on the other hand, by definition, come straight from the patient tumor and are therefore far more physiologically relevant. The major con to using primary cells, however, is that generally they can only be cultured for short periods of time. Believe it or not, the success rate for establishing a new cell line is still relatively low, even though these are cancer cells. Therefore, you'd have to keep procuring tumor tissue or freshly isolated cancer cells regularly to continue your primary cell studies. One emerging cell model type that bridges the gap between these cancer cell lines and primary cells are known as the cancer organoid model, or tumoroids. These are freshly patient-derived models that are grown in 3D and may be cultured for a relatively long period of time as compared to primary cells grown in 2D. The culture method for these can be relatively difficult, but it may be worth it for your research if you desire high physiological relevance. One final note: rather than choosing one or the other, these can be used in tandem or sequentially, with your research starting with cell lines to narrow the scope, then moving to primary cell culture later for later studies, for instance. And tumoroids are not the only cells that can be cultured in 3D, of course. Cell lines can also be cultured in 3D, either fully encapsulated in a basement membrane extract like Geltrex or Major Gel, or in suspension. But should you culture your cells in 3D? Well, it depends. Both 2D and 3D culture have pros and cons. 2D culture is more widely utilized as it is the easier of the two culture methods. It has a larger publication record with which to contextualize one's research and can be easier to monitor for response to experimental conditions by imaging, for instance. However, because adherent cell lines grow in 2D in a monolayer, the cells are experiencing a decidedly non-physiological environment. They are attached to a plastic substrate that is much stiffer than tissues in the body. The cells are only interacting with each other at the edges in 2 dimensions. And all the cells have free access to nutrients in cell culture medium. Cells in 3D, on the other hand, have 3-dimensional cell-cell interactions, do not interact much, if at all, with a relatively stiff substrate. And the cells will have different access to the nutrients from the medium depending on where they are located within the spheroid of cancer cells. However, 3D cultures are notoriously difficult to dissociate and often utilize specialized protocols and equipment for imaging. Therefore, depending on your research question, you may or may not want to undertake 3D culture, or you may want to start with 2D culture and progress to 3D at a later point. Of course, paramount to any of this research is the type of medium that you are feeding your cells. It is very important that you think through both the type of the basal medium that you would like to use, as well as the supplements that you are planning on using. And it really depends on your cell model and your research question. The scientific literature is a good place to start to find a medium, but you may want to adjust it for your specific experiments. For instance, if you are utilizing estrogen receptor positive breast cancer cell lines, you may want to use phenol red free basal medium, since phenol red is a weak estrogen mimic. Similarly, a specialty fetal bovine serum, namely charcoal stripped FBS, which has been treated to remove hormones, may also be useful for estrogen related studies. Similarly, you must carefully choose the culture vessel or cell culture plastics that are right for your purpose. First, consider if you need your cells to adhere or not. This will help you choose between non-treated or tissue culture treated vessels. Of course, you will also need to consider if you are just scaling up your cultures or if you are performing high throughput experiments. If the latter, high throughput, meaning number of conditions or replicates, etc., do you need? Also, which type of assays will you be utilizing? Are you looking at fluorescence? You may want black wall plates. Are you growing spheroids? You may want to try very low attachment plates like Nuclon Sphera. And cell culture insert plate systems have a variety of uses that we'll get into in just a little bit. Now back to our hallmarks of cancer. Since we've gone over the cell models and tools you may need for your cancer research, let's discuss how you can model angiogenesis, invasion and metastasis, and deregulating cellular energetics in vitro, starting with angiogenesis. Angiogenesis refers to the growth of new blood vessels. This is key for cancer progression as tumors can only grow so big without being vascularized. If a tumor does not have its own blood supply running throughout, the cells at the center of the tumor will start to die as nutrients will not be able to reach them. Therefore, as seen in the video, when there is a small tumor forming or breaking off from the primary site, it will secrete angiogenic growth factors like VEGF in order to attract endothelial cells to proliferate and infiltrate into the tumor, thus allowing it to continue to grow. Angiogenesis is also key for another hallmark of cancer, metastasis, as the tumor cells utilize blood vessels to migrate from the primary site to distant tissues for colonization. Therefore, cancer cell endothelial cell interactions are very important to study. These types of studies can be done with cell culture in several ways. For modeling angiogenesis in vitro, endothelial cells must be cultured with an extracellular matrix mimic or basement membrane extract like Geltrex in order to form tube-like structures and networks. If you'd like to study the impact of secreted factor crosstalk between endothelial cells and cancer cells, this can be accomplished with co-culture and cell culture inserts. One could also look at more direct contact by embedding endothelial cells and cancer cells together and into an ECM mimic. More complex models may incorporate the spatial relationship between cancer cells within a tissue and endothelial cells lining a patent vessel, which is best modeled with microfluidic devices, wherein fluid flow reminiscent of blood flow can also be accomplished. Although angiogenesis and metastasis can be observed in these complex microfluidic devices, transendothelial migration can also be observed in a simpler setup. And we'll discuss that now. As cancer progresses, cancer cells can invade the local environment near the primary tumor site, and in some cases migrate across the endothelial cell barrier into the blood vessels in a process called extravasation. Once inside the vasculature, the cancer cells can be carried to distant sites far from the primary tumor and extravasate or cross the endothelial cell lining again, once more to colonize that distant site and form a secondary tumor. The process of intravasation, extravasation, and colonization is called metastasis. In the video, you can see cancer cells break away from the extracellular matrix of the primary tumor site, intravasate, and extravasate to the secondary tumor site. Invasion and metastasis can be studied with cell culture in several ways. One of the simplest methods is a wound healing assay, where you scratch a monolayer of cells with a pipette tip and see how long it takes to close the gap in the monolayer. The rate at which the wound closes is related to how migratory or invasive the cells are, either due to the cell type or the condition to which they were exposed. In the figure below, we show the increased wound healing response of breast cancer cells with exposure to heat-stable basic FGF as compared to a low serum medium condition. A far more complex way to investigate invasion and metastasis is with microfluidic devices, wherein endothelial cells can be seeded to line a channel within a cancer cell-laden extracellular matrix mimic, and cancer cells can be observed extravasating within the device. A classic and more widely used method for modeling invasion, however, utilizes cell culture inserts. So let's take a closer look at this approach. Here we have diagrams showing the classic setup for migration and invasion experiments. For those that may not be familiar with them, cell culture inserts are essentially smaller wells that fit inside the larger wells of a well plate. This forms an upper chamber and a lower chamber where media conditions or cells can be seated separately, but can also interact. The interaction can occur because the cell culture inserts have a porous membrane for a bottom instead of solid plastic. Therefore, for migration experiments, cancer cells can be seated in the upper chamber, and a soluble factor that attracts the cancer cells to migrate towards it, known as a chemoattractant, can be added to the bottom chamber. If the cells are invasive or metastatic, they will migrate through the pores to the underside of the membrane, where the number of migratory cells can be counted. For invasion experiments, the setup is identical, but before the cells are seated onto the porous membrane, the membrane is first coated in a basement membrane extract like Geltrex. This adds yet another barrier that invasive or metastatic cells must cross to reach the chemoattractant on the other side of the membrane. So let's look at an example of this in action. For this experiment, non-metastatic MCF7 cells and highly metastatic MDA and B231 breast cancer cell lines were used. The chemoattractant was 10% FBS, which was added only to the bottom chamber. In the top chamber, the cells were seated in serum-free medium. After 20 hours, the cells on the underside of the porous membrane were counted, and the percent of cells that migrated was calculated. You can see that in both the migration setup without Geltrex and the invasion setup with Geltrex, a much higher percentage of the metastatic MDA MB231 cells cross the membrane in response to FBS as compared to the non-metastatic MCF7 cell line. Also interesting is that for the migration setup, close to 90% of the MDA MB231 cells made it across the membrane, versus only about 70% that made it across in the invasion setup. This demonstrates the additional barrier to migration that the Geltrex coating provides. So now that we understand the basics of modeling cancer cell endothelial cell interactions and invasion and metastasis in the lab, let's take a look at a more complex example. With this experiment, an extravasation event is modeled. If you look at the panes of the figure from left to right, the model gets more and more complex. In panel A, you just have the metastatic prostate cell line PC3. The cells are stained blue and that are seated in the upper chamber and the same medium seated above and below the membrane. As there is no chemoattractant in the bottom chamber, there is little migration. Next, in panel B, serum-containing medium was put into the bottom chamber. And just like in the previous example, the serum induced the PC3 cells to migrate across the membrane. In panel C, instead of serum-containing medium in the bottom chamber, bone cells known as osteocytes were seated in the bottom, and their secreted factors acted as a chemoattractant to induce the PC3 cells to migrate across the membrane. Finally, in panel D, you have the full extravasation model. Endothelial cells, which are stained red, are seated on the underside of the membrane to form a barrier representative of the wall of a blood vessel. Again, the osteocytes are used as the chemoattractant, and amazingly, you can see by the blue on top of the red in the bottom image that not only do the PC3 cells migrate across the membrane, but also across the endothelial cell monolayer in response to the soluble factors that the osteocytes are secreting. This experiment is also a fantastic example of how cell culture inserts can be used for co-culture experiments with a variety of cell types. The final hallmark of cancer we will discuss today is deregulating cellular energetics. This is a fairly complex topic, but in short, most normal cells and tissues utilize mitochondria to get energy and only switch to a secondary method of metabolism known as glycolysis if oxygen is in low supply. because glycolysis is a relatively inefficient way to get energy. Well, in the 1920s, Otto Warburg and colleagues observed that cancer cells were using far more glucose than the surrounding normal cells. They realized that cancer cells often utilize glycolysis to meet the high energy requirements for tumor growth, even in oxygen-rich environments. This phenomenon was termed the Warburg effect, and it is a true hallmark of cancer. The increased glucose uptake of cancer cells is even used in clinical imaging to diagnose cancer and monitor tumor response to therapy. Cancer cell metabolism not only has implications for tumor growth, but also response to anti-cancer drugs, and is therefore very important to model in a physiologically relevant way in vitro. One tool that is very exciting to accomplish this is human plasma-like medium, or HPLM. HPLM was modeled after human plasma and therefore is more physiologically relevant than traditional cell culture medium. For instance, regarding the glucose, which was so critical to the Warburg effect, HPLM has about a third of the concentration of glucose of other traditional media formulations. As you might imagine, this can have a major impact on the metabolism of cancer cells and their response to anti-cancer drugs. In the data shown here, you can see that it took about 175 times the amount of the cytotoxic compound staurosporine to kill the same number of cells in traditional DMEM medium as in HPLM medium. This may be explained by the unique metabolic profile that cells in HPLM have versus traditional media, as shown in this next slide. For this study, we have spheroid models of 3 different cancer cell lines. Each line was grown in 3 different conditions: a traditional cell culture medium plus regular FBS, HPLM plus regular FBS, and HPLM plus dialyzed FBS. You can see by the bar graphs that depending on the media formulation and the type of serum chosen, the cancer cell metabolism as measured by ATP production is significantly different. Therefore, it is very important to consider the formulation in which you are growing or assaying your cells. The closer we can match an in vivo-like environment to study cancer in the lab, the better our in vitro results will hold up when translated from the lab bench to the bedside. So that wraps up our overview of how to model in vitro 3 of the hallmarks of cancer outlined by Hanahan and Weinberg. Hopefully this will inspire you to delve into studying one or more of these hallmarks in your own research. Cancer is so common, so complex, and such a difficult problem to solve. We can certainly use all the help we can get to better prevent and treat this awful disease in the years to come. For more information on the hallmarks of cancer, how to choose your cancer cell models and culture method, please check out our Cancer Cell Culture Basics Handbook at thermofisher.com/cancer cell culture. We also have a wealth of other valuable information there, including cancer cell culture protocols and our Cell Culture Heroes seminar series, which is wonderful. And I invite you to check it out. I really appreciate your attendance and attention today, and I'll be happy to take any questions we have from the audience. Thank you.
Thank you, Brittany, for providing such wonderful insights on her cancer cell research. We'll now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, on your slide on invasion and metastasis, I saw that heat-stable basic FGF was used. Why is heat-stable basic FGF chosen rather than native basic FGF?
Yeah, that's a great question. So native human FGF BFGF, rapidly loses bioactivity at standard culture conditions, like 37 degrees Celsius. This means that more frequent media changes and more protein is required to maintain biological activity. Now, heat-stable FGF is specially engineered to be thermostable and to retain bioactivity at physiological temperatures. It has more than 90% sequence homology to the native protein, but it has greater than 80% bioactivity after 72 hours at 37 degrees C. So it's a very cool product. Highly recommend heat stable BFGF.
Okay. Thank you. The next question we have just came through is, which cancer cell lines can I grow in Gibco human plasma-like medium?
Yeah, also a very good question. So we've tested and shown compatibility of HPLM with a variety of cancer cell lines representing many different cancer types, breast cancer cell lines like MCF7 and MD8 and B231, of course, HeLa cells, LNCAP, A549, and even JERCAT cells. You use the HPLM just like you would your typical basal medium. You supplement it with FBS, and then you're good to go. You don't have to adapt it from your normal medium at all. Even beyond cancer, we've shown that primary natural killer cells can also be grown in HPLM. So if you're interested in cancer immune co-culture studies, you can utilize HPLM there as well. We do have a full list of all the cancer cell lines and other cell types that we've tested on our website.
Great, thank you. And looks like we have time for just one more question here. I've noticed that Thermo Fisher offers a variety of cell culture inserts for invasion and migration studies. How do I choose which one to order?
Yeah, you have to be careful. So the biggest difference in the cell culture inserts is the pore size. So different applications and cell types require different pore sizes. The first step is to determine what pore size you need for your study of interest. If it's a smaller pore size, like 0.4 microns, this would be for applications where you don't want the cells to migrate through the pores. So this would be just for co-culture or transport type studies where just secreted factors are going above and below the membrane. The larger pore sizes, like 8 microns, it's what's recommended for studies where you would like to see the cells migrate, chemotaxis, invasion studies. I would recommend checking out the application notes on our website, and that should help guide you into which cell culture insert product would be appropriate for your study of interest.
Okay. Well, this wraps up our Gibco Galaxy Tour webinar today. I would like to thank Brittany for sharing her cell culture experience in cancer research, which I'm sure will be very beneficial to all of the cancer cell researchers. A big thank you to our audience for joining us today and submitting interesting questions. For those questions that we were unable to answer today, we will follow up with an answer via email. We hope all of you have gained more knowledge today that will help you enhance your lab skills. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to ask any and everyone to take our survey at the end of this webinar, just so you can tell us your feedback. When you complete the survey, you will be entered into a drawing to win a Gibco exclusive Lego set and a featured product from this webinar. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's lab event. Hope to see you again for our next Gibco Galaxy Tour webinar focusing on genome editing in October. Thank you, and goodbye for now.